Now we will study another interesting type of microscopes, which is called two-photon microscopy. You can see in the figure, it is really very compact and complex type of microscope, and it will be based on the photons microscopy. Two-photon microscopy is a fluorescence imaging technique that allows images to living tissues up to about 1 millimeter in depth. Before we were studying the confocal microscope, we were able to see the images which were less than 100 millimeters in length or 100 millimeters in depth. Now we will see about 1 millimeter in depth the two-photon microscopes. The basic principle lies behind the two-photon microscopy is the specimens are stained with the fluorochrome. Two-photon microscopy uses long wavelength, the red light, and therefore the two photons instead of one, and are needed to excite the fluorochrome to emit the lights. As we have already studied the confocal microscopy and the fluorescence microscopy, all of these three microscopes, these are used the fluorochromes in their microscopic material. Specimens are stained with the fluorochrome, and two-photon uses the long wavelength light, which is used in red light, and therefore the two photons instead of the ones, and are needed to excite the fluorochrome, and which will emit the light. The longer wavelength allows the images of living cells in the tissues up to 1 millimeter, which will and up to 1000 nm deep. Just see in the slides the focal plane, one of the on the left side and one is on the right side. The single photon emission which will shown on the left side, and similarly the two photon emission is much clearer and much better as compared to the single photon emission. The two photon microscopic illustrations in the figure, the infrared lasers and scanning mirror on the left side, and the dichroic beam splitter. Green PTM will be coming from the left side, and it will be reflected by the dichroic beam splitter. If the light or some of the dichroic beam splitters which will go through, and it will be again stopped by the dichroic splitter, and it will be make it clear with the object which will be below the dichroic beam splitter. Similarly, the red filter and the green filter will be on right side, and the lights will be coming from there, and the fluorophores and the focal plane will be make it more clear the object. The two photon microscopy's applications, it is most of the time used for the thin cells, which are the skin cells, which are very thin and transparent cells, or it will also be used for the invasive optical biopsy. As clear in the slides which has been shown, the two photon excitation microscopy and measures corpuscles are very clear, and the application of the two photon microscopies are clear from this kind of slides.